Okay, thank you very much for having us. I would like to thank ARM for that, and also thank you all for attending. I will be making forward-looking statements today, and I encourage you to look at the risk factors in our 10K. Okay, so if you're not familiar with Fibrocell, we focus on creating therapies around the autologous fibroblast, and we're targeting skin and connective tissue diseases. And we're looking both at cell and gene therapy applications for scarring diseases, but also rare diseases as well. And we're focused now on 3 indications. The first is a cell therapy approach for chronic dysphonia, which is a condition of loss of voice due to scarring of the vocal folds. This is a phase 2 program that's actually based on a BLA we already have approved back in 2011 in the field of aesthetics. The next 2 indications listed are gene-modified cell approaches. We're working with the Intrexon Corporation in rare diseases. The first, FCX007, is for just a very devastating blistering disease known as recessive dystrophic epidermolysis bullosa. It's a long title, RDEB, but again, it's a congenital orphan skin disease, mainly affecting children. We do have orphan status for this, and also we have rare pediatric disease designation from the FDA. FCX013 is our next gene therapy candidate that we're targeting in Linearscleroderma, and I'll talk more on these conditions as we go through. So I went ahead. Okay, so talk a little bit about autologous fibroblasts. Fibroblasts, I'm sure you're all very familiar. They're the cells that produce the extracellular matrix in tissues and are involved in wound healing, and the most common cell in connective tissue. So they're ideal candidates for diseases where we target skin and, again, connective tissue. So we see some key advantages with our approach, and there's been some discussion on panels today just recently about this, but this is a monogenic disorders are what we target. With no mechanism actions, and I'll talk specifically to that on RDEB. The fibroblasts are genetically modified ex vivo, so we actually have safety testing on the cells off before we administer to the patient. We've gone through and designed multiple assays to characterize these cells, so we have really well-known product attributes. We inject locally, so we're delivering these into the low vascularized tissue of the skin right to the area of concern around blisters. And we have reduced rejection concern from the autologous nature of our product. And of course, these are based around the IP we have for use and method around our proprietary technology for manufacturing. So here's a graphic of the 2 platforms that I just talked about. On the left is the cell therapy platform that we already have approved through BLA and we're using for our phase 2 program in dysphonia. Simply, the cells are taken from biopsies collected from tissue from the patient. They're sent to our manufacturing facility in Exton, Pennsylvania, which is right outside of Philadelphia. We expand the fibroblasts in vitro, low passage process. They're cryopreserved, and then when we're ready for administration, the cells are pulled, washed, thawed, and then shipped overnight to the site. The right side talks about the work we have in Intrexon, and obviously we're using a viral vector here. We're using lentivirus. It's a self-inactivating lentivirus that we package our target gene constructs, and then we introduce them through transduction to the cells in early passage. And then obviously it's an integrating virus. We integrate the construct, and then we expand those through the rest of the process, and then it follows the same path as our cell therapy. So obviously we brought a known cell therapy process that's already been vetted through the regulatory process of the BLA, and then now merging that with Intrexon's synthetic DNA technology. So this is an overview of our platform. Obviously, our most advanced program is chronic dysphonia, which is in phase 2. And our other 2 programs are in preclinical with RDEB, the more advanced, moving into phase 1. And I'll talk a little bit about our timelines coming up. So first on the cell therapy front, chronic dysphonia, if you're not familiar with the condition, simply it's a causing of an issue from scarring in the vocal fold. What you see on the left is a healthy vocal fold through a scope. On the right is a heavily scarred condition, and what happens is you lose the vibratory pattern of the vocal fold, so essentially you have a diminished voice, or in extreme situations you have a loss of voice. As this occurs either idiopathically, it can occur over age, it can also occur from surgeries and from other conditions as well. Current treatment for this is really just voice therapy. There are surgeries, and also there are some other products that have been tried, such as fillers, fillers and implants, but not with much success. We look at the prevalence of this condition to be about 125,000 people. We have run a phase 1 in this. It was published in the Linearscleroderma. It's an N of 5 trial, so it was pilot, but all the patients completed. We didn't have any serious adverse events, and we saw sustained improvement using the measurement tools from 3 to 12 months. So that moved us into a phase 2 study, which is currently fully enrolled. We're at over 20 patients. It is a double-blind, 2-to-1 randomized placebo-controlled study with a 4-month endpoint, and we expect to have dosing done by the end of the year and primary endpoint reported in the second quarter of 2016. So now I want to move on to our gene therapy programs. The first, of course, is RDEB. RDEB is a monogenic disease. It's caused by a mutation in the COL7A1 gene, which encodes a protein known as type 7 collagen. The reason this is important for this disease is type 7 collagen actually makes up what's called an anchoring fibril, and I'll show you a graphic in a moment, but the anchoring fibril binds the 2 layers of skin together, and when that's not present and the fiber is not present or is not synthesized by the patient, you actually get these 2 layers of skin floating on top of one another, and a little bit of lateral friction actually separates them and causes blisters, and then when there's pressure on the blisters, it starts laterally growing. So it's obviously a very, very painful condition. FCX07, of course, is a autologous human dermal fibroblast that we're transducing with lentivirus in hopes to replace the protein. And here's a small graphic just to sort of illustrate this. Those green fibrils that are moving from the dermal epidermal junction down the dermal layer are the anchoring fibrils, and again, when they aren't present, you get this separation of the 2 layers of skin. It's a very painful disease. They're diagnosed at infancy. They actually see the trauma at infancy, and there's a high mortality rate. Actually, one of the largest problems is squamous cell carcinoma with all the wounding and turnover, and of course, sepsis. There are no treatments for this disease. They're generally just wound healing attempts using bandaging and antibiotics. It has a very high economic impact. There are families reporting paying more than $10,000 a month just simply to care for wounds on their children. And there's a lot of surgeries. They deal with feeding tubes because of malnutrition. The blisters also occur in the oral mucosa, and surgeries, including hand surgeries and also esophageal dilatations. And we estimate there's about 1,100 to 2,500, so it is an ultra-rare population. And so talk a little, we presented data at ESHG in Glasgow back in the summer, and we were presenting some of our in vitro protein characterization data. And if you look at the slide here, collagen 7 is formed by 3, most collagens, obviously all collagens are trimeric, but they're formed by 3 290 kilodalton alpha chains that form a trimer. And it's important because that's essential for the functional construction of the final anchoring fibril. So we wanted to characterize that using multiple assays. The first, obviously we want to show we can synthesize it. So in the upper right in A, you see the results of a dose-dependent, so basically different viral high and low dose of the lentivirus COL71 construct. So you can see it is dose-dependent on the viral titer, and we're producing collagen 7. And the bottom immunoprecipitation Western blot basically shows that we are producing the 900 kilodalton fully formed trimeric form. So just making alpha chains isn't enough for functionality. You do need to have that trimeric structure. So we showed that as well. So the conclusions from the poster are listed here. I review the majority of them here. But the take home is, obviously at full scale GMP manufacturing, we produced collagen 7, we produced it at the proper size and structure, and then second from the bottom there, we also produced a number of characterization assays to show it binds to other proteins in the skin, including laminin 332, which is essential for its functionality to complete the anchoring fibril, and we also showed some migration assays that make them look like more normal cells instead of RDEB phenotypes. And, just to have an ultimate, high amount of characterizations of these cells, and we obviously replicated that over multiple autologous lots to show consistency. So that poster is posted on our website if you're interested in seeing any more. And actually at this very moment at the American Society of Human Genetics, we're presenting a poster on our preclinical data. So just for a quick summary, for toxicology, we ran a hybrid farm tox study where we took skid mice and we grafted them with human skin grafts and then injected the product into that graft. And after 2 and 6 weeks, we saw no adverse conditions, we saw no tumors, we saw no change in blood chemistry statistically from the baseline and from the placebo group, and we saw no vector distribution after performing qPCR on the various organs in the mouse. And for proof of concept, we took RDEB skin substitutes we grew in vitro, and then those were grafted on the mice, and then we were actually injected the product, and then we look at immunofluorescence. We can actually tag and look at a human tagged anti-Col7 and look at not only the production of collagen 7, but also that it accumulated in the epidermal junction. And why that's important is that's the actual location where the anchoring fibril must form, and we saw it not only produce, but we see it migrate to the right area. So if you do follow our story, you know we submitted these studies to FDA for review, and we were asked by FDA, as we recently announced, to perform another supplemental tox study, this time in a non-grafted mouse model, SCID mouse model, which we're now gearing up to execute. We're going to amend our IND that was filed with the new toxicology data in the first quarter of 2012, and we expect to initiate our phase 1/2 trial in the second quarter of 2016. So I want to reiterate that there wasn't any known toxicology issues. We were just asked to supplement the package. So here's a real quick summary of our phase 1/2 trial. Again, it's a rare disease, so we're looking at treating 9 patients, 3 adults, and then based on the results of the initial 3 adults, we're looking to move into a pediatric population. Obviously, we're evaluating the safety of FCX007, but we're also going to be looking at a mechanism of action endpoint, which we will be looking not only at immunofluorescence for the anti-Col7 marker and biopsies collected from the treated area, but we also be looking at an electron microscope analysis to actually look at the morphology of the anchoring fibril, which is important as well. And then we'll be using 3D photo imaging to actually monitor the wound healing of these of the wounds and look at the reepithelialization rates as part of an early efficacy analysis as well. So again, looking to initiate this second quarter of 16. Okay, the final indication, which is our earlier preclinical that we're moving on, is Linearscleroderma. Linearscleroderma is also known as a class of diseases known as localized scleroderma. These are an autoimmune disease that causes a reaction in the skin to hyperproduce collagen 1 and 3 that results in these fibrotic lesions on the body. And in the linear form, they're these long, sort of almost look like a hypertrophic scar, but these long lesions that can, appear anywhere, but where they become dangerous is when they appear on joints. Not only can it cause pain and reduce range of motion, but they're common in children, and if they form on joints early and aren't treated, they actually can cause developmental issues. And they've been known to happen over joints, which can cause development issue of the joint. As you see in this picture, there's one leg has the scar in the inner part, and then the other leg is normal. Or they can occur on the forehead, which actually can lead to cranial disformities and neurological conditions. Right now, current treatment is corticosteroids, typical what you would do for an autoimmune, physical therapy. We think there's about 40,000 patients who have this condition over joints that would qualify. So where we are in this development program, we've developed the product profile, we demonstrated protein expression. This will be a gene-modified fibroblast that is encoded with a protein to actually degrade the excess collagen production, and it'll also be under an inducible switch control, promoter switch control, that is a proprietary technology from Intrexon called REO-switch, and that'll be integrated as well. So now we've got the gene identified, we target it. We're preparing actually now a scale-up manufacturing batch for this to start our preclinical work. Our animal models have been vetted with our KOLs, and we're got that in development. So we're anticipating having proof of concept ready by the end of the first half of 16, with an idea with a target to file the IND for this in the end of the year, fourth quarter of 16. So in summary, we have a lot of milestones coming up in 2016 we think are very exciting. Between the trial initiation for 007, proof of concept data, and IND submission for 13, and back on the cell therapy front, we'll be reporting top-line data on our phase 2 program for vocal fold scarring and chronic dysphonia. The summary of our financials are below, and as you can see, we're well capitalized, and we look forward to reporting out more and more data and sharing it with everybody in the future. Okay, thank you very much. I appreciate your time.